Anna Shirazian.
My name is Ardeep Singh.
I'm Lizette Wisa.
And I'm Karina Sarabia.
And we're going to be talking to you about convergent evolution, possibly seen in bioluminescent squid. Our mentor is Sabrina Pankey. The faculty advisor is Todd Oakley. We're with the Department of Marine Biology, Ecology, and Evolution with the JKCB program. Alright, so convergent evolution. What is convergent evolution? Well, here's a pretty good example. Here we have a picture of a fly and a bird. They both have the ability to fly, and they both have wings, but since they're very distantly related, we find that very interesting. So what probably happened was some sort of similar natural event or occurrence that caused them both to have the ability to fly, which is basically what is convergent evolution. The process whereby organisms that are not very closely related develop similar traits independently.
So you may be asking yourself, why is evolution so important to our project? Well, I think we're all familiar with the theory of evolution, but really understanding the basics of evolution would allow us to make progress in biological sciences. And as you saw in the example with the bird and the fly, we're going to apply the same concept of convergent evolution to these very different types of squid.
So for our project, we're working on two different squids, and we're looking for gene expression in bioluminescence. The one over there is Eurotethus edulis, and it is found near Japan. This one is Euprymna scolps, and it is found near Hawaii. You might be thinking they're both squid, but the thing is they have been set apart by 300 million years of evolution. Very different evolution from each other. But what is interesting to us is that they both have developed the same bioluminescent trait, and we want to know what gene could be playing a role in that bioluminescent trait.
Another interesting thing is that these two squids not only developed bioluminescence, but they developed it for the same reason, as a defense mechanism. Envision yourselves at the bottom of the ocean looking up, and you see the silhouette of the moon. A squid that is not bioluminescent swims by, and what you would see is their shadow. Well, if this squid was bioluminescent, instead what you would see were just bits and pieces of its shadow, hiding it from its predators. So what we're trying to do is we're trying to quantify gene expression. Well, what is gene expression? Gene expression is what makes cells different. Take, for example, these two cells. This is a human liver cell, and these are human skin cells. They both contain the same DNA. But what makes them different is the parts of the DNA that they use or they express.
So for our research, we're looking at three main genes that we wanted to quantify. The first gene is responsible for making the protein actin. Actin was our positive control protein. It is present in all cells. The next gene was responsible for making opsin. Opsin is sensitive to light, and it is present in Euprymna and Eurotethus. The last gene that we looked at is responsible for making the protein lopopsin. It is similar to opsin, but it is only present in Euprymna.
So in this 3D structure, we have lopopsin mapped onto opsin, and in the blue we see opsin and white is lopopsin. And what we find really interesting about this 3D structure is that these two proteins are very different. They evolved very, very differently, and the fact that they're still similarly structured means that they could be related in light perception, which could lead to similarities in the bioluminescence gene.
So when we're going to try and quantify the gene expression in different tissues, we're going to do it in the eye of the squid, which is this green area, the brain, the pink area, the photophore. The photophore is the pouch where these squid hold their bioluminescent bacteria, in the skin, and in the gill. The gill is going to be our control because we do not expect to find bioluminescent genes expressed in the gill.
So quantifying the gene expression in the squid, it's a process with many steps. So in the first step, we isolate the genetic content from the squid, genetic content also known as messenger RNA. And then in the second step, we use this enzyme called reverse transcriptase to make a copy of the DNA, which is called complementary DNA. And in the next step, we're going to quantify the abundance of the gene using a machine called real-time polymerase chain reaction, or known as real-time PCR machine.
Okay, so a brief introduction on how a PCR machine works. First, you have your double-stranded DNA in your reaction vessel. The DNA is then heated up, and this is in order for the DNA strand to uncoil and for these hydrogen bonds to break. So once the hydrogen bonds break, the DNA becomes single-stranded. When the DNA is single-stranded, this allows for a primer to attach to the DNA. So now we're going to have a primer go ahead and hybridize the DNA molecule. When the primer hybridizes, it allows for an enzyme to attach to the single-stranded DNA. When the enzyme attaches, it'll be able to make more copies of the DNA. So in just a second, the temperature is heating up and the enzyme attaches. And now the enzyme is going to proceed and start making copies of the DNA. When the DNA becomes double-stranded, our special dye called cybergreen will be attaching to the DNA. And when the DNA is double-stranded and is expressing this fluorescence by the cybergreen dye, the PCR machine can go ahead and take a picture using its camera and start quantifying the gene levels that our reaction vessel has. So the more DNA there is, there's more fluorescence, and the PCR machine is able to pick this up and quantify the data.
And the last step of this process is to analyze the statistical data.
Okay, so this is a graph that we got from the PCR machine, from the real-time PCR. This is the quantity of actin expressed in the gills in the Euprymna bulbs. So the first thing I want you guys to see is the cycle. The PCR machine could be set to any amount of cycles. We said for our experiment we only needed 40 cycles. Every cycle the DNA doubles, and in 40 cycles we had enough information where we could relate how much gene is present in where. On the left side is the fluorescence level. This is where when the DNA doubles, the machine takes a picture every time it doubles. So this line right here, this is known as the threshold level. This is just an arbitrary line to where we can compare two different genes and the quantity of them. So this is the curve for actin. If you're wondering why we have two lines, it's just because we took two samples just in case one went bad or something happened, we would have a backup. So from the threshold line, we can find out that 22.4 cycle, this is where we're going to compare the actin to the opsin, which can be seen right here. And we also took two samples just in case one went bad. And the cycle for opsin was 32.9. So the difference between these two cycles is only 10.5, but you have to think that every cycle the DNA is doubling. So if you want to think about that in numbers, in the gill, there is 1,448 more actin molecules than opsin, which is what we expected because gill was our control. We didn't expect that much opsin there to be in the first place.
Okay, this is a graph of our data. This graph is a little bit different. It is actually a log scale graph, which means that every number is actually the exponent. So for example, down here we have 0. That's actually 10 to the 0, or 10 to the first power, 10 to the second power, and so on. Right here on the horizontal axis, we have the tissues that we sampled from. And so our reference point was the photophore because we found the least amount of opsin in the photophore. And so what that means is that for every 1 molecule of opsin in the photophore, we found 95.4 molecules of opsin in the skin of Euprymna. And in the skin of Euroteuthis, for every 1 molecule of photophore, we didn't find any. In the brain, we had a problem with the sample of Euprymna. It didn't produce any data that we could analyze. Something could have gone wrong with the DNA or anything like that. So we weren't able to use it. For Euroteuthis, though, we didn't find any opsin in there. In the eye of Euprymna, we found a great deal of opsin. We found that for every 1 molecule of fluorophore, we found 11,936.5 molecules of opsin. In Euroteuthis, we found that for 1 molecule of opsin in the photophore, there are 10.6 in the eye. And for 1 molecule of photophore, In the gill of Euprymna, there are 13.3, and we didn't find any for Eurotubus. This graph is similar to the previous one, except that this is for lofopsin in the tissues versus lofopsin in the skin. Here our reference point is the skin because we found the least amount of lofopsin in the skin compared to the other tissues. So, like I said, in the brain we didn't have any data for that. In the eye we didn't detect any lofopsin. But in the gill, we found that for every 1 molecule of lofopsin in the skin, there's approximately 1.8 molecules of lofopsin in the gill. And for photophore, for every 1 molecule of lofopsin in the skin, there are approximately 19.6 molecules in photophore.
Okay, so our conclusions. Our results weren't entirely what we expected, so we do think that we need to run more tests. For one, Euretitas had little to no expression of opsin in the eye, which is very odd considering that most eukaryotic animals have opsin in their eye for light perception. So the fact that we didn't see that probably means that our DNA sample, there was something wrong. We need to take another sample and run more tests. And also, it could be that we don't really have the right idea about opsin being responsible for bioluminescence. But as we said previously with the diagram with the lofopsin and the opsin superimposed on each other, there is research strongly suggesting that there is some sort of relationship between opsin and bioluminescence. So we need to do more research and really see what's happening. The Euprymna sample, brain sample, did not yield any data, meaning that there was some sort of problem when we were doing the reaction. It could have been that the sample was contaminated, so we need to run another test and try another sample of DNA. So future experiments, like I said, we need to do further testing to see really what opsin's function is in bioluminescence. And we need to take more samples from different squids, different species, and run more tests and see really what's happening. Okay, so we would like to thank our mentor Sabrina Pankey for letting us work in her lab and helping us out, the faculty advisor Todd Oakley, our RA Jason Murphy for helping us out on the slides, the Department of Ecology, Evolution, and Marine Biology for having us, and the Jack Kent Cooke Foundation for funding our research and stuff, and everybody who has come to see us, thank you very much.